Our next talk is from a scientist who is working to help provide that hope. So I will welcome Dr. Qinglan Ling to put her video on. She's going to present the design and the preliminary data on a novel gene therapy strategy developed for individuals with MTATP6-Leigh syndrome caused by a specific mutation. So I'll pass it over to her.
All right, good morning, everyone. And first, I'd like to thank Lauren for sharing her story. That's such an amazing story. And I think that's the reason why we're all here, is to help the kids and the family like her. And I'm currently assistant professor at the Umass Medical School, and I joined this beloved Leigh syndrome community 7 years ago. And I just cannot ever say that I'm going to stop what I am doing, no matter how challenging it is, because of all the patients and the families here. And so thank you, Cure Mito Foundation, for giving me this opportunity to present what my team has done in developing gene therapy for MTATP6-Leigh syndrome. Let's see. All right. So our audience here all know that mitochondria play an important role in the cells, and where a critical component of mitochondria is the electron transport chain that locates in the inner mitochondrial membrane. And it is composed of 5 complexes, as indicated here, the complex 1, 2, 3, and 4. And the last complex is called complex 5, or ATP synthase. So 99% of the components are proteins encoded in the nuclear DNA, such as SIRF1, which is synthesized in the cytosol and then translocated into the mitochondria. And there are also certain critical subunits that are encoded directly from the mitochondrial DNA, such as MTATP6, which is the main topic of today. And so the primary mission of my lab is to develop gene therapies for primary mitochondrial diseases, especially electron transport chain disorders and diseases that are associated with these mitochondrial dysfunctions. So MTATP6 encodes the ATP6 protein. It is directly transcribed and translated within the mitochondria. It forms a critical subunit of complex 5, or ATP synthase. And the function of ATP6 is to facilitate proton transport to drive the rotation of the serine and the ATP synthesis. And the most common mutation of MTATP6 is a 993T2G mutation, which changes the wild-type form of leucine into arginine, and also A993T2C mutation, which changes the wild-type leucine into a proline. And this amino acid is highly conserved. So when we talk about the mitochondrial DNA, we need to bring up the concept of heteroplasmy, which means the presence of both wild-type and the mutant mitochondrial genome within a single cell. So for MTATP6, in our cell, we can have a mix of wild-type MTATP6 gene as well as a mutant MTATP6 gene. And as a matter of fact, as we all get older, and we all carry certain mutations in our cells in the mitochondrial DNA. So if you have a low content of mutant copy, that means we have a low heteroplasmy, which will lead to a later onset and less severe symptoms. And the patient with low heteroplasmy normally develop a NARP, or neuropathy ataxia, and retinitis pigmentosa. However, if you have too many mutated mitochondria MTATP6 copy number, then we call it high heteroplasmy. So those patients, they show an earlier onset and more severe symptoms, and this tend to develop as Leigh syndrome symptoms. when I first started looking into this disease, I was thinking about developing more straightforward gene replacement therapy using allotopic expression. Basically, what this does is that we will use a adeno-associated viral vector to deliver a functional copy of the MTATP6 gene and we add a mitochondrial targeting sequence at the N-terminal, so that when the protein is synthesized in the cytosol, then it can then be translocated into mitochondria, whereas in the nature, the protein is synthesized within the mitochondria. So we did a little bit engineering here, so then I try to test this strategy in the cell, as we can see here by using the immunofluorescence to determine the location of this engineered protein. That the green represents the protein that we express, and the red means indicates the mitochondria. And if we look at the merged image here, if we look at the merged image here, we can see some colocalization, but not a high percentage. That means that the protein that we express was translocated towards mitochondria. So then we want to know whether this protein was integrated into the inner mitochondrial membrane, because that's where it will perform its function. So here we used a proteinase K assay, and we first isolated mitochondria from the transfected cells. And then we performed a proteinase K digestion on this mitochondria. And what proteinase K does is that it'll digest the outer membrane of the mitochondria while leaving the inner membrane and the matrix intact. So then we performed a regular Western blot to examine the protein expression. And I want everyone to focus on this red circle here, that as we can see in the isolated mitochondria portion, the HSP60 indicating the matrix integrity, that with or without proteinase K, we can see the about even expression of this protein, suggesting that our proteinase K treatment did not digest the inner mitochondrial membrane. However, if we look at the outer membrane marker TOM20, that with the proteinase K, the intensity of this band significantly went down. That means the outer membrane was mostly degraded. What's most important is the middle here that we are looking at, the ATP6 expression. So as we can see, without the proteinase K, we were able to see a pretty decent band, but after adding the proteinase K, meaning digesting the outer membrane, then the band is almost gone. So that means that our protein did not integrate into the inner mitochondrial membrane. So then we sort out another more novel therapeutic approach by using the mitochondrial targeted cytosine base editing method developed by Dr. David Liu. And what this does is that it uses two DNA construct that can perform a CC to a CT editing. And I'm just highlighting this this part here that we can see the DDDA11 was able to change this C8, C9 to TNT. So this is what we're using to perform the gene editing. And what we're doing there is to leverage that technology to develop a base editor that can target two pathogenic variants, meaning the A993 T2G and also A993 T2C. So for the A993 T2C, we can just use this base editor to revert to the wild type 4 by performing a C editing to a T, and then we can change that proline to a leucine. However, for A993 T2G, as we can see, this is a transversion mutation, and then this base editor technology cannot help us to revert it back to the T. So what we propose to do is to do a functional restoration where instead of editing this mutated G here, we're editing the C right next to this G, and then we are changing it to a T so that we then substitute this amino acid from arginine into a tryptophan. So this does not change back to the Y-type leucine, but it's changing to a tryptophan to restore the function of the protein. And this hypothesis was supported in a previous study that performed in the East, where I want you to just focus on this last column of numbers here, that if we have a Y-type, then we have 100% of ATP synthesis. If we mutate it to arginine, this is a A993T2G mutation, then the synthesis went down to 10%. And then however, if we substitute the arginine to a tryptophan, then the synthesis went up to 83%. So we think that because of this, if we can perform this successfully, perform this editing, then we can potentially restore the function of ATP synthesis. So what we did in the last couple of years is that we designed and generated 18 pairs of this mitochondrial base editing. And then after testing on the cells, we determined that 2 out of 18 pairs of this base editors showed a positive editing as shown here. Although this editing efficiency is not very high, as we can see, the highest here is about 5%. And so we're still keeping trying to optimizing this. But when we tested the function of this editor, as we can see here, if we replace the glucose with galactose in the culture media, so that we're forcing the cells to only use mitochondria as the main energy source instead of glycolysis. And then by looking at these two columns here, that with this base editor, we were able to show not much, but significantly 20% in cell growth increase compared with EGFP control cells. And then we also use the immunofluorescence to determine the location of this base editor. As we can see here for the left construct, the protein 100% co-localized with a mitochondrial marker. And however, for the right construct, it wasn't as perfect as we wanted. So our next step is also to further optimize this so that we can increase the editing efficiency. And yeah, so I kind of went through a little bit of the future directions, but what we are doing now is to optimizing the choice of mitochondrial targeting sequence. And also we want to design zinc finger binding domain instead of using the tail DNA binding domain to increase the on-target editing. And also we can potentially fit both construct into one AAV. And we're also trying to sort out novel approaches for delivery and to regulate the base editor expression level after they are delivered into the cell. So with that, I would like to thank my team members and all my colleagues at the Umass Medical School to help us make this possible. I've only been here for a year, and I think it's great that everyone is so collaborative here. And this project was started when I was a postdoc at UD Southwestern with Dr. Stephen Gray, as well as my co-mentor, Dr. Prashant Mishra, and I really appreciate their guidance. And this project has also been funded by Uplifting Athletes, Cure Mito, as well as NIH. And I really appreciate that. And last, I just want to thank all the support from Lee syndrome family and the patients. Thank you.
Thank you so much, Qinglan, for sharing how you're so carefully navigating the challenges of these novel mitochondrial gene editing techniques and how you're gathering data towards human intervention. So I think the next talk follows really well from that.